simple but powerful model system, Caenorhabditis elegans. It is important to know how this 1 millimeter creature has led the research for the last 30 years or so and paved the way to discover the constant pathways that regulate lifespan in multicellular organisms. These animals are transparent. As you can see on this slide, the C. elegans hermaphrodite and males are noted separately. They are transparent and easily visualized, like it's easy to visualize them under the stereo microscope with liver, bone, intestine, and body wall muscle. With an average mean lifespan of 16 days, these animals are extremely useful for aging research. As you can see in the video, the physiological changes and body morphological defects get very obvious with age in these animals. So what is aging? Why scientists are investing so much of their time to understand this biological process called aging? So aging is thought to be an increased rate in fragility that leads to an increased mortality rate as a function of time. It is also often thought as a non-specific degeneration outcome as a result of accumulation of damage from environmental exposure or metabolic process. So with aging being inevitable in almost all organisms, it is now in the interest of biomedical research to explore the conserved mechanism of protein function underlying this important biological process. Thus, my research question is to understand how the master scaffolding protein Pry1, which is a functional homolog of axin in mammalian system, regulates lifespan in C. elegans. So axins are the negative regulator of the well-studied Wnt signaling cascade that has been studied for the last 20 years or so and is important in regulating multiple developmental events in organisms ranging from sponges to vertebrates. So axins are the negative regulator that forms a destruction complex and inhibits the Wnt signaling cascade. So when you have axin, the destruction complex is active and the pathway is off. In the absence of axin, there is a constitutive activation of the pathway that leads to various diseases including cancer. So axins are important regulators of multiple biological events that includes cell proliferation, differentiation, and body axis patterning. Consistent with such an important role of axin, it has been shown to be associated with diseases like axis duplication and neoectodermal defect. My research has so far provided the first evidence of this master scaffolding protein being involved in lipid homeostasis. Thus, understanding the regulation of this protein and its signaling cascade promises to accelerate the development of tools and methods to modulate this protein during adult-specific or aging-related diseases. So in my project, I have been trying to characterize the role of this master scaffolding protein axin. But since the last 1.5 years or so, I focused my research only on the adult-specific role of this protein. As you can see from the reported analysis, this gene is actually expressed in multiple different tissues and at all the larval stages. So the tissues include hypodermis, muscle, intestine, and neurons. But what I am interested in is the function that Pry1 relies on for regulating lifespan in the adulthood. In the adult-specific expression, you can see that Pry1 is found to be expressed strongly in the muscle, the hypodermis, and the neurons. So to address the tissue-specific requirement of this protein in maintaining lifespan, I used the RNAi interference system in C. elegans and knocked down this protein only during adulthood in hypodermis, muscles, and intestine. Interestingly, in the hypodermis and muscle, after lowering Pry1 function, the animals showed a significant reduction in lifespan. But to figure out whether this function is adult specific or whether the primal function in regulating muscle health and lifespan relies on its function only during development or the adult specific stage, I overexpress primal in the hypodermis and muscle using tissue-specific promoter. But interestingly enough, muscle-specific promoter extends lifespan even after the overexpression of primal during the developmental stage, which is opposite to the phenotype that has been seen in the hypodermis. So if you overexpress primal, Wnt signaling is turned off or it perturbs the Wnt signaling cascade. But if you overexpress primal no matter at which stage of animal, it is beneficial and it extends lifespan, as you can see in the figure panel B. Next, to understand whether this extension in lifespan seen in the animals having overexpressed primal in the muscle, I also analyzed whether the